project is on sex-linked differences in early onset Alzheimer's disease. This topic caught our attention because early onset Alzheimer's disease has been proven to be more frequent in women rather than in men. We will test this hypothesis by using genotype tissue expressions tool of a sex-linked analysis of APP, PSEN1, PSEN2, and GRMS5. Through this, we hope to answer a question to see if Alzheimer's disease is based on sex. We hypothesize that the genes amyloid beta precursor protein APP, preselin-1 PSEN1, and preselin-2 PSEN2 are more prevalent in women, while glutamine metatropic receptor 5, GRMS5, is more prevalent in men. Some background on this disease is Alzheimer's disease is a neurodegenerative disease that is both complex and multifactorial. Of the total number of individuals with dementia, 60 to 80% of them have Alzheimer's disease. Alzheimer's disease can be classified into two general types, early onset and late onset. Early onset Alzheimer's disease occurs in individuals under 60 years old, while late onset Alzheimer's disease occurs in individuals older than 60 years old. Early onset Alzheimer's disease is less common than late onset Alzheimer's disease, accounting for 5 to 10% of cases. A small number of these can be attributed to single gene mutations in the APP, PSEN1, and PSEN2 genes, with the formation of amyloid beta plaques of neurofibrillary tangles, also known as tau pathology, in the brain, and individuals can be neuropathological diagnosed with Alzheimer's disease. The average lifespan of an individual with Alzheimer's disease after diagnosis is 4 to 8 years. Although some individuals can survive upwards of 20 years, the known risk factors of AD Alzheimer's disease are age, sex, and genetic differences. It is also known that the prevalence of AD in women is much higher than in men. Women account for 2/3 of AD cases, with the estimated lifetime risk for AD Americans at age 45 or 65 being 20% in women and 10% in men. The first step of our methodology is to isolate the genes in the brain by using the genotype tissue expressions tool to filter results by sex as well by tissue affected. This would be the brain, hippocampus, and brain frontal complexes genes. Next, we use NCBI's genetic testing registry GTR to further isolate the genes and to identify their biological context. From this, we found that APP, PSEN1, PSEN2, and GRMS5 are higher in men than in women. Looking further into these findings, APP in women's frontal complex was 538.6 TPM and hippocampus of 273.7 TPM. And then for men, the frontal complex was 580.8 TPM and hippocampus 293.1 TPM. This pattern repeats for the rest of the genes to conclude that there is a difference in genes in men compared to women. Our graphs show a side-by-side comparison of the bulk tissue gene expression for each gene in men compared to women. Females are pink and males are blue in our graphs. From this study, we found that the biological context of the genes allowed us to identify why the numbers were higher for men than to women. APP codes for a cell surface receptor and transmembrane precursor protein that forms peptides. Some of the peptides have antimicrobial properties and promote transcriptional activation. When a single gene mutation occurs, it is implicated in autosomal dominant AD, APP, PSEN1, and PSEN2 mutations regulate the production, aggregation, and degradation of beta amyloid peptides, the primary component of amyloid plaques. GRMS5 codes for metabotropic glutamate receptor, which signals the activation of the phosphatidyl calcium messenger system. This system is involved in the regulation of synaptic plasticity and neural network activity. Some treatments for Alzheimer's disease utilize the GRMS5 gene to treat AD, although this treatment is more effective in men rather than to women. We hypothesized that the large numbers of APP, PSEN1, and PSEN2 genes would increase the likelihood of AD. This is not true. We concluded this is because the increased likelihood of AD is due to the mutations occurring in the genes rather than the number of them. In conclusion, from the study, we found that our hypothesis was wrong. Although men have more of all four genes, higher numbers do not equate to higher numbers of mutations. With early onset AD, what matters is the mutations and not the amount of genes present. In hindsight, what we should have done is compare the mutated genes in men and women to see if there were differences in the numbers and if this could potentially be a link to the increase of Alzheimer's disease. Overall, the extensive research and execution of genotype tissue expressions tool of a sex-linked analysis of 4 genes associated with Alzheimer's disease answered our question of can certain genes be a direct link to an increase of Alzheimer's disease based on one's sex. The strength of our research would be we directly answered our hypothesis with the data we use and the genes we chose to do an analysis of. A weakness would be we did not answer the overall question of why Alzheimer's disease is more prevalent in women rather to men. But our analysis did discover sex-differentiated effects on gene expression with the result that higher numbers do not equate to higher numbers of mutations. These findings contribute to the big picture, the biological explanation for the prevalence of Alzheimer's disease in women being much higher than in men. By using novel diagnostic tools, we can help contribute to future research of Alzheimer's disease.